Hello everyone, today we are studying a very important topic. The topic name is two component system. So this is present in bacteria. It is for the signaling. See, we having sense organ, by that we can know what are the things are surrounded of us, right? We can see, we can hear, we can touch. By that only we come to know what are the things are there, right? But in the bacteria, they don't have any kind of eye, ear, and any sense organ. So how they will know where they have to move, where how they have to take food? If suppose food is little far from it, how it will go toward the food? It cannot see, right? So they having a very important signaling pathway, that name is two component system. Okay. And what happening in this? See, before this is in the bacteria, what happening? I just draw the structure. See, this is the cell wall of bacteria, and cell wall of bacteria, it having one receptor. This is the receptor. Some part are grown toward the, some part are in the environment, and some are in the cytosolic phase. Okay, so what happening? Bacteria release attractant. What it is releasing? It release attractant. Suppose this is the attractant, and this attractant, what happening? It bind to the ligand. Okay, so this is the attractant, and if suppose sugar and any other molecules are present there, so this attractant will bind to the bind with the sugar and amino acid. Okay, and this complex will come near to the receptor. It bind to the receptor, and when it bind to the receptor, when this complex, attractant and ligand complex, bind to the receptor, this receptor get excited, it phosphorylate itself. Autophosphorylation is happening at the place of histidine. Okay, this is his kinase. What it is? It is his kinase. So this protein name is his kinase, and this his kinase get excited. Sorry, it phosphorylated. It phosphorylated at the histidine place. Wherever histidine is present in his kinase, it will excited, it phosphorylated. Okay, now when this phosphorylated, when this his kinase is phosphorylated, it phosphorylate another kind of protein that containing aspartate. That protein containing its soluble cytosolic site, it present in cytosol, and soluble protein that phosphorylate, that phosphorylate. Now this aspartate get phosphorylate by the his kinase, by the excited his kinase. Okay, so now what happening? This soluble protein will go near to the flagella. Suppose this is the, this here is the flagella. Okay, this is the flagella. So now this soluble protein will go near to the flagella, motor protein, and now it will excite the protein that present at the motor side of the flagella. Okay, and it phosphorylate. Phosphorylation of flagellum. So it phosphorylated. Sorry. So it phosphorylated, and phosphorylation of flagella. Now this flagella is excited and it start movement. Okay, it start their motion, and when it start movement, it will go where the all attractant and ligands are coming. That complex is coming, it go toward, when it will go nearer to the attractant ligand complex, more attractant will bind to the receptor, right? When it will go away from the food, then definitely the receptor will not bind to the complex. Okay, so by that way they are knowing they now they are reaching toward the food and it moving away from the food. So this is the way. Okay, so bacteria actually, I mentioned some points. Bacteria release attractant in environment. Ligand is what? Any kind of sugar and amino acid. Okay, so ligand attractant bind to the receptor domain of the membrane. The first component, see why the name is two component, because the first component is the hiskinase that autophosphorylate when the ligand and attractant bind to it. And second component is soluble protein, that is also called response regulator. What this is the very important point. One is the hiskinase, another is the response regulator. So this response regulator will excite spartate, and the excited phosphor now this after this phosphorylation, it phosphorylate vessel protein of the flagella. Okay, now flagella is excited and it will start movement and it will go toward the food. If more attractant is binding to receptor, it means it's reaching toward the food. If less number of attractant binding to receptor, it means it is moving away from the. Okay, so see the structure of this. This is the receptor and this is the hiskinase. So it phosphorylated by the ATP, and when it phosphorylate another protein that containing aspartate. This is called response regulator, and by this protein it excite the flagellum. Okay, so this is the all two component system. And by when the flagellum protein is excited, it phosphorylated, then it start movement and it will go toward the food. So see, this bacteria is moving toward the food when more number of attractant bind to receptor. Okay, and same with the toxic also. When the surrounding if having the toxin, what will happen? It will move away from the toxin. Okay, because if they will bind, definitely they will die, right? So this is about the two component system of bacteria. I hope you understood this all. If okay, thank you.